Welcome to my diary. Today we are going to be discussing glycolysis and fermentation. In glycolysis, glucose, NAD+, and ADP are converted into pyruvate, NADH, and ATP. It's also important to note that this takes place in the cytoplasm of the cell. We begin with a molecule of glucose, which is phosphorylated by ATP into glucose 6-phosphate. Glucose 6-phosphate is then phosphorylated by ATP again to produce fructose 1,6-biphosphate. At this point, this is broken down into two glyceraldehyde 3-phosphates. For now, we're just going to take a look at one of them. This is phosphorylated by a phosphate compound, such as hydrogen phosphate, and NAD+ at this point is reduced into NADH, an important electron carrier. The ADPs are phosphorylated to produce ATP, resulting in pyruvate. The same thing happens for the other molecule, so this results in 4 ATP. Since 2 ATP was consumed in the beginning, the net ATP production is 2, and 2 NADHs are produced. Now let's investigate lactic acid and ethanol fermentation, which also takes place in the cytoplasm. Lactic acid takes place in animals, while ethanol usually takes place in yeast and plants. Let's begin with lactic acid fermentation. We begin with pyruvate, which was produced during glycolysis. NADH, which was also produced during glycolysis, arrives, resulting in the production of lactic acid and NAD+, which can be recycled again in glycolysis. In alcohol fermentation, we begin with a molecule of pyruvate. However, a carbon is lost in the form of CO2, resulting in the formation of acetaldehyde. NADH is oxidized into NAD+, which can be recycled in glycolysis to produce ATP as needed. Thank you for watching.